Well, I guess Dwayne started our really discussion in multiple ways, but I'll first just mention my disclosures here. But really, Dwayne in the morning mentioned the really detailed introduction of the B cell responses. So I'm not going to spend time on that, but I'll mention a few things that are relevant to the data that I'm going to present. So first is when you get immunized or infected, actually the first responding subset is of B cells that really we can detect in blood, the plasmablasts. These are the cells that terminally differentiated that produce all the antibodies that we can really measure by ELISA or by neutralization very early on after infection or vaccination. But we have been studying what happens in blood for decades. What I'm going to discuss is really what happens in the draining lymph node. And as Dwayne mentioned in the morning, that this is the site for a very unique reaction for B cell responses. It's the site for the germinal center reaction. And in this reaction, affinity-based selection of among the responding B cells for those that actually bind to the antigen with a tight affinity, and those are the ones that selected to become memory B cells. And those are the ones that become also gradually become long-lived plasma cells. And those are the cells that maintain our serum antibody titers for extended periods of time, long after those plasmablasts, the very early wave, have already died. So in my talk today, I'm going to discuss in detail the germinal center B cell responses to the SARS-CoV-2 vaccine. Primarily, I'm using the mRNA-based vaccine. It's actually Pfizer vaccine. And one thing I would, just because the note that Dwayne just mentioned, yeah, I think one thing is that we learned that immune responses in humans take much longer time. One of the things that I find very surprising and that I'm going to present to you today, that that reaction, while those plasmablasts disappear from blood within a week or two max after the last immunization, that reaction has been lasting for 6 months so far and still ongoing in many individuals. And I'll show you that this is something that I find exceptionally amazing. And then I'm going to show you at the end what that means for that particular compartment, bone marrow. This is, are we actually seeing the graduates that going from the germinal center now going to become bone marrow plasma cells? Okay, so when I first started my own lab here at WashU 4 years ago, I really wanted to do this, to access the draining lymph nodes in humans, because I knew that this is where you can dig deep and learn about how, why, and at the time I started studying immune responses to influenza vaccine, and I really wanted, what's defective with this vaccine? Why it's suboptimal? What can we do to improve it? And looking at blood was at certain point wasn't really very informative for that particular vaccination. So I really wanted to dig deeper and see what happens in the draining lymph node after we get vaccine. Very simple question: do we actually induce a germinal center response? We know that for successful vaccine, you have to have memory B cells, long-lived plasma cells. But are we actually inducing a germinal center response after this vaccine to actually get those subsets? So I teamed up with an excellent radiologist here at WashU and the School of Medicine, and I mentioned that can we actually access axillary lymph nodes in humans using fine needle aspirates before and after vaccination? And so it's a process that sounds more scarier than actually it is. It's relatively simple, at least for radiologists, not for me. But basically, this is what you're looking at is by ultrasound. This is a draining lymph node. This is an axillary draining lymph node. In that case, it was from left axilla. And then the needle, you can see it here going. This is the cortex of the node. This is the region. So this is the cortical region. This is where the germinal center would be. And you can see that the needle goes in. We try to sample only the cortex. And we do this by, we call it 6 passes. And then I take the needle and put it in media, and then obviously do all of the immunological downstream analysis on that. And we can do this repeatedly on the same node over time. And we have actually now, for some participants, we have done this up to 16 times in the same node without noticing any defect in terms of the quality of the response over time. And I'm not going to show these data, but this is something just in case I get asked about this frequently. So how the data looks. So if you basically look, this is the node of before vaccination. This will be an axillary node. You'll see that the cortex is very thin, relatively, because this is non-reactive node in a way. But then after vaccination, this is after influenza vaccination, you get really nice thickening from the cortex over time. And then when we actually analyze the cells that comes out of those nodes, so at day 0, at baseline, you don't see any germinal center B cells. And we have verified that if you look in the lymph node, again, you can really look in the details this is all published, so I'm not going to go into details, but we verified that this subset is indeed the germinal center B cells. This is the CD20 high 38 intermediate in the draining lymph node. And within this germinal center response, we can detect hemagglutinin specific, which is the main molecule or main protein in the vaccine. Over time, the antigen-specific responses within this node. So this told us very important thing. So this is with the first evidence of already a vaccine-induced germinal center B cell response in humans, especially with a vaccine that, as we know in the US, it's a vaccine that's giving to 160, 170 million people a year. It's non-adjuvanted vaccine, so it's really not a very potent one. But indeed, in some individuals, we get this really nice germinal center that actually continued for 8 weeks, which was very encouraging for us. But then it allowed us to ask an important question about, are there any difference between the clones that are recruited to the germinal center reaction versus those that are recruiting to the very early plasmablast response, the one that is short-lived? We know that those are the clones that are destined to become memory B cells and long-lived plasma cells. So these are the kind of questions we were able to answer with this kind of approach. So I'll just show you that if you compare, we made monoclonal antibodies, as Dwayne discussed. The same technique as we single cell sorted the germinal center B cells, but also the plasmablasts from blood that was isolated early on, compared them in terms of clonal overlap, and then also compared the level of mutations. And what we learned from that is that usually the memory B cells would be actually able to participate not only in the early plasmablast response. Remember, in influenza, we all have this pre-existing memory cells, but can also participate to become germinal center cells. But then we found a new subset of cells that almost coming from naive B cells, they have much lower somatic harbor mutation compared to the plasmablast or the shared clones. And those cells only came later in the germinal center and recognized novel epitopes in the vaccine. So we realized that the germinal center, in a way, becomes a window to recruit novel B cells or naive B cells that haven't been engaged before. And I think this is very important for us to realize that even if you get multiple immunization, and there is nothing we are more immune to compared to influenza, I can tell you that. I'll show you data for that. We are hyper immune to influenza in a way, and even in that case, our germinal center is still able to pick, even if it's a few naive B cells, to re-engage them or engage them actually in that case in the response. And this is depicted in this. And as I said, the details for that is really have been published. So I went through this fast just to introduce you to the technique. So this was where we are, or actually where we were just before the pandemic. And then the pandemic hit. Obviously, all our influenza studies everything was shut down, and we had to, we were fortunate enough that we have that infrastructure set up to actually apply it for studying immune responses to SARS-CoV-2 vaccination. So this is the WashU 368 study that I'm running here at WashU. And that study, actually, it recruited literally the first 40 plus people that got the immunization, mostly all healthcare workers here at WashU and the Barnes and Jewish Hospital here at WashU. And by chance, 7 of them had confirmed SARS-CoV-2 infection. Again, they are emergency medicine workers in ER, and so there were exposure at the time. We started in last December. We collected blood before vaccination and multiple time points after first and second vaccination. This is all Pfizer. We collected lymph node from the same site where they got the vaccine. For one individual, we collected lymph node. That person got the vaccine in different arms, but we'll discuss that if we have time. But also, we added the bone marrow compartment because I really wanted to see if we can see the transition between germinal center and bone marrow in those individuals after vaccination. So So that's the layout of the study. We are at the 6 month after second vaccination time point, like everyone else at this stage. So this is showing the binding. As you know, there's nothing surprising. People that never got the exposure, they started from baseline. This is binding to the spike protein, and I'm showing only here the IgG. And you can see that after first dose, there's some increase, much better increase after the second immunization that given in 3 weeks, and then the slow decay starts. I'm going to show you the more kinetic, more long-term decay later. But as many groups have shown before, that if you have been primed by infection before, you obviously don't start at baseline, but you get a really nice boost after the first dose, and then the second dose adds almost nothing. But then you start seeing the decay after that. And neutralization actually makes a difference. Having the infection before, this is neutralization against the wild type virus. Having infection before definitely gives you a really nice boost in terms of neutralizing antibodies compared to just getting the vaccination. But this was done at the peak of the response. At later time points, most likely this will even out. Although probably having the infection before would still have you an advantage. I have to say that. So now, how about this is all coming from, as you now understand, that this is all coming from the plasmablast response, because this is the short term. So what happens in the germinal center? And this is where really what determines the durability of the response, what determines the quality of the response. And so one thing that my radiology colleagues told me right away, because we were doing the influenza studies right before the COVID vaccine. And with influenza, we always struggled in a way to find the right lymph node. And this is something I didn't mention, but finding the nodes is actually the hardest part, because there's 30 to 40 nodes in the axillary region in humans. And only a few of them actually drain the upper arm area. Most of them actually drain parts that are the breast area, the back of the neck, areas that has nothing to do with our vaccine input. So we learned this the hard way, actually. And then one of the things that we were hoping for is to be able to find the nodes, because influenza, it's not really easy to find it all of the time here. What my radiology colleagues told me after the very first subject was enrolled, they told me, oh, the nodes are easy to see. And not only that, they are exuberant. And for someone who's not a native speaker, this is not something that I use frequently as a term. So what they meant by that is that the node was very juicy and huge. And they told me it's very vascular and very reactive node. Obviously, all of this was, for me, there was a good response. That's all what translates to. And I really wanted to see what that means in terms of germinal center response, obviously. So what that translated to is that this is what we see. This is our best flu responder. So we are looking at a BCL6 intracellular staining and 38 intermediate isotype switch B cells. So this is by definition germinal center B cells. Before vaccination, there's nothing, almost some background or something going on. But then after vaccination, this is 12 days. This is for us was huge. This is our first 4 subjects in the mRNA vaccination. This is the germinal center response in those individuals. And this was obviously very robust, and it really obviously mirrored by the lymph nodes. But one thing that was even more remarkable with this is that we saw this multiple lymph nodes involved. So this is one example from one person that we saw this germinal center being huge for a long time, but then we see a second node, especially after the second vaccination, we see a second node, and then we started sampling the second node, and we still see huge germinal center. And in both of them, they are NG-specific responses. But then what even more remarkable that I see this, and this is based on studies that I've been doing animal studies before we moved to the human part. We never saw a germinal center response continue at that high active level for almost 6 months. And as you can see here, and it is not only just based on activation marker on germinal center B cells, but based on spike binding. So this is after first vaccination. This is 1 week after second vaccination. And then we are at 6 months, and some individuals still ongoing, and those individuals will be offered the booster, as we're going to discuss. So for me, this is remarkable. This means that the reaction is still going on. We are still putting cells out, potentially putting cells out for becoming memory. Probably there are memory B cells coming out all the time. And this explains in a way our studies, or many studies that came out, that the memory B cell frequency is increasing over time. And that's really not surprising seeing this data. Now the question becomes what this means for long-lived responses or long-term antibody responses. And for that, we went, and this is just to mention, this is a sample for one individual, but this is a summary for multiple individuals with multiple lymph nodes. So this is total GC, this is total spike-specific GC, and in a way, this is measured as a spike binding germinal center as a percent of CD19 in the node. And in a way, you can argue that this is even not going down. It's starting to go down in some individuals, but we are actually seeing that some are really doing well in terms of their immune response or ongoing immune response in that case. So what this means for the long-lived plasma cells, these are the cells that are become obviously terminally differentiated. They take a residence in the bone marrow and responsible for maintaining the serum antibody responses. So first we looked at the serum antibody responses. So not surprisingly, very consistent with many other groups that have already published this work. This is not published, but this has already been shown by many other groups that if you look at the IgG, and again, black here is the people that have been exposed to the virus before vaccination, and red is the people that haven't no evidence of exposure before. And you can tell that there is an advantage, obviously, for people that had been exposed. And this is not surprising. This basically they had 6 months heads up with an infection before getting the 2 doses, so they are definitely had an advantage. So it's not really surprising. Obviously, they had an advantage on IgA, and that's again not very surprising. But the fact that we see an antibody response at that long time, this definitely a really indirect evidence, or probably almost a direct, that these antibodies are being produced by long-lived plasma cells. So can we now look at those? So as I mentioned, I'm going to show you the bone marrow plasma cell frequencies in some of those individuals at that time point. And the way we do that is we actually, obviously, we use the iliac crest to sample the bone marrow. We purify the CD138 plasma cells. These are the bona fide plasma cell markers, and then we do an ELISpot assay. It's a very simple assay where you just coat your ELISpot plate with either the spike protein of the SARS-CoV-2 or with actually flu vaccine. And in that case, we used influenza vaccine, we used tetanus vaccine as control because everyone had been exposed to those antigens. So we wanted to see that they have a long-lived plasma cell compartment. And here's the data you can see from an example for one individual. Here we're looking at flu vaccine. And as I told you, every one of us looks like this. We have a lot of plasma cells against influenza antigens. And blue here is the IgG, and red is the IgA. This will be the total plasma cells. This is the one influenza. This is how the tetanus diphtheria looks. This is using the vaccine, so much less than influenza. And to a similar level, this is taking six months after second immunization. We see the spike antibody responses in this case. Obviously, we have seen the spike before. There was nothing, and then they started coming up, and that's what we are now. And this is the summary for the old individuals we tested so far for this. So you can see that the spike is obviously much less than influenza, slightly less than those against the tetanus, but we definitely see an established compartment, which is really not surprising at this stage, showing that how persistent the germinal center. So what we have learned so far is that mild SARS-CoV-2 infection primes for a more robust and durable actually B cell response against SARS-CoV-2 vaccination. And the problem with infection is the outcome is really variable. You always see when you see the titers after infection, there's much more variation compared to vaccination, but by no means that infection wouldn't be a really good prime for vaccination. And this has been shown by many groups. We show here that something that I find really surprising, given all what we learned. I mean, what we have learned so far about germinal center response was based on animal models, and really what we learned here is that it really takes the reaction much longer. It will be important to really understand what really controls its durability, but really means, do we really need to add more antigen? Do we need to add wood to the fire? The fire is still going, and it's really what will happen when you add more antigen to that. That's something that I'm always wondering about. Do we really, maybe it would have been, I agree with Wayne, it could have been better to actually space out the vaccines than 3 weeks, especially seeing these kind of immune responses, that maybe if we let the Germinal Center after the first immunization kind of mature and then give them the antigen again, that would have been probably have a much better outcome. And we are seeing some signs of that after AstraZeneca from Europe, the spacing part. And then I showed you the bone marrow part, which is this is unpublished, that this is part that we are really, in a way, really establishes that we have a really good vaccine that that not only is strongly immunogenic but can protect against, and showing much really good protection against not only the circulating variant but also, well, the previously circulating variant, but also against the emerging variants. So with that, I would like to acknowledge first the multiple heroes in my lab. To be honest, in the last 20 months, they've been working hard on many other studies that I didn't have a chance to present. There's a lot of unsung heroes in this part. As you can imagine, if to do these kind of studies, given that I'm not, and I don't have any clinical training, and you know, this requires a lot, not only wonderful colleagues, but dedicated colleagues to be able to get these studies on. Even the study coordinators here really played a major role to be able to get blood, lymph node, and bone marrow from the same individual on the same day, talking to 3 different departments, 3 different rooms, 3 different buildings. It's a massive work. I wouldn't be able to do it without the collaboration. Obviously, the volunteers. This is not something that you get easily. We'll forever be grateful for them, and obviously the funding. And I'm happy to take any questions.
Thank you, Ali, for the great talk. I'll remind panelists and attendees, please raise your hand. We'd like to make this as interactive as possible. So if you post a question in the Q&A, it'd be great if you could raise your hand and read it. I did want to start out with a relatively simple question, and I see Steve has his hand up already. When you looked at the persistent spike-specific germinal center B cells at the 6-month time point, it looked like there were a handful of patients where there was a large decline. And I'm just wondering if you could comment on the nature of what's going on in those folks.
Yeah, so it's interesting because and some of those individuals I mentioned, there are a couple of nodes at least. Some actually we see up to 3 nodes going on. The reaction is going on, especially after the second dose. And we see in one node the things starting to slow down, and then by 6 months it's crashing. But then the other node is still going strong. So I have absolutely no idea what that means. I mean, both nodes have been sampled, you know, same time or all time. But one of them is just the reaction was concluded. We are now doing the deep dive into these, you know, the different clonal types and all of that to try to, are they actually the same B cell clones in both compartments, both nodes, is one is ahead of the other by any means? It's really fascinating for us to be able to even start looking into that. But I have really no idea why some persist longer than others.
Go ahead, Steve.
So I was wondering, what do we think about the need for that, you know, increasing response over such long times? Does that require continued spike? Do you think the spike is still being made in some way, or is it long gone? And nevertheless, the response gets better and better, even without spike.
I guess I'll be saying I don't know a lot today, but it's a beautiful question. I don't think the spike is being made at that stage. I think it's unknown, and since one of the important gaps we have at the moment, how long like spike is being made after that vaccination. I would doubt that the mRNA would be still around past week or 2. So I think that the spike could be present. We know that from the germinal center in the form of immune complexes that are deposited on the FDCs, the follicular dendritic cells within the germinal center. So that's probably what keeps things going. The question is, what really why we don't see this after influenza, for example. So is it how much of that complex being deposited, or how, or the affinity of the cells as we go on are increasing? So we're really depleting these complexes that are deposited. And it just it's one of those things that we would like to do, and it might be hard to address in humans very precisely. But I think the question about how long the mRNA persist, how long the spike is presented in a non-immune complex form would be great to answer.
Yeah, or can presented peptides continue to be presented for a long, long time? I guess we don't know the lifetime of these things. Yeah.
Yes, not yet. I'm sure there will be a lot of studies on that in the next few years.
Anyway, thanks.
And I have to ask, are you following these studies up with studies on the other vaccines that are available?
Well, the problem with, obviously the only one available for us here in the US is J&J, and the problem with that is it started later, but we started looking. It was harder to recruit for it. We do have 12 individuals who got the J&J. We haven't done the detailed analysis. We looked at the lymph node, and we do see germinal center that's specific for the spike in some of those individuals. So we are doing it. It's basically almost 3 months behind in a way. So we will 3 months behind. So we started these studies in March, so we will be getting the 6 months time point very soon.
That will be interesting, because you could imagine that the expression would continue for months. I mean, the DNA might persist in the adenos, and it might make spike longer. Yeah.
There are some serological data that came actually from Harvard, actually showing that the responses seems to be better persisting after adenovirus, and also from AstraZeneca, some data from Europe as well.